Hi, everybody. Let's talk about the cell cycle. The cell cycle is a way that cells basically divide. It's how cells go through their life cycle. There's two basic kinds of cells that exist on our planet. There are prokaryotic cells and there are eukaryotic cells. And the main difference between prokaryotic cells and eukaryotic cells is that prokaryotic cells are smaller and they don't have a nucleus. Eukaryotic cells are larger and they do have a nucleus. Along with that nucleus, those eukaryotic cells also have a lot of different specialized machinery inside of the cell. Prokaryotes, also known as bacteria, their only special machinery are ribosomes. And I know that you know what ribosomes are for, so I won't bore you in talking about them. Anyway, the replication process or cell division process that occurs in prokaryotic cells is much simpler than eukaryotic cell division. No matter what the process is, though, the process has to begin with some kind of DNA replication. Now, the DNA replication that we've been talking about in our class is very similar in these two different cell types. Bacteria have helicase, they have DNA polymerase, and they use those enzymes in order to make molecules of DNA that are identical to each other. Basically, that whole semi-conservative replication process applies to bacteria, just as they do to our own cells. Now, the coolest thing about bacterial cell division is that we get daughter cells that are genetically identical to each other, as well as to whatever the cell was at the start. This kind of cell division is basically cloning. It's cloning because there's no genetic variation introduced in this process. Now, I guess there's the potential for genetic variation, a variety of genes coming up, but the only way that you get variation in this particular kind of a population is just through mutation, which again, we all know what mutations are. Now, when we're talking about eukaryotic cell division, there's two different types. There is mitosis, and mitosis is what we're going to be focusing on today. Mitosis is a special kind of cell division where we get daughter cells that are identical to each other. It's kind of like cloning in human form, I guess. The other kind of cell division that we'll talk about later on is meiosis, but that's not for our topic for today. That's for our next unit. Our cells go through the process of mitosis in order for our body to grow. In order to add new cells to our body, think about a fertilized egg. A long time ago, we were all just a single cell, and that cell went through mitotic cell division, and then there were two cells. Those two cells copied their DNA and divided, and then there were four cells. Those four cells copied their DNA and then divided, and then there were 8 cells. And on and on and on until we get a teenager. Now, if you're a single-celled complex organism, something like a paramecium or euglena, this might actually be the way that you reproduce. You might go through the process of mitosis to make more of your population. However, this is not how humans reproduce. Once again, when humans are going to go through the reproduction process, we go through a special kind of cell division to make our gametes, our eggs, and our sperm. And that process is known as meiosis. When our cells are getting ready to go through their cell division, and after they've copied their DNA, they basically try to organize that DNA into structures known as chromosomes. When a cell's not dividing, when it's just living its life, when it's going through and making proteins, making messenger RNA, going through transcription and translation, the DNA is in a unique form that we call chromatin. And what chromatin is like, it's basically like just a stringy mess. Think about if you took a spool of thread and you unwound it. You'd just have this, collection of thread, but there wouldn't be any organization to it. And that's all right when the cell's just going about its daily activities and not dividing. However, when the cell is going through some kind of cell division, different machinery inside the cell work in order to make it so that the DNA twists up, it folds, it condenses, and it tightly coils to form this very unique chromosome structure. Now, this is how I see it. My own personal analogy. I think of chromatin as being like my clothes that I'm going to take on vacation. I don't just show up at the airport with my arms full of my t-shirts and my socks. I'd probably drop something along the way and my clothes wouldn't make it on my flight. Instead, I neatly fold up all my clothes and I pack them into a suitcase. So I see suitcases and chromosomes as being really similar. A chromosome is a structure where the DNA is all packed up inside of it, all nice and organized, ready for cell division. Here's some other images that hopefully can help you get a better feel of the relationship between chromatin and a chromosome. Over here on the right, we have DNA, and this DNA is typically wrapped around certain kinds of proteins that are called histones. And this mess over here on the right is what we have in a cell when the cell is just living its life and when it's not going through any kind of cell division. This would be DNA in chromatin form. And if we're using a microscope to view a cell and its DNA is in chromatin form, we typically can't even see the DNA inside of the nucleus because that DNA is so thin. It's not wide enough to be visible to our eyes, even though we are using a microscope. Now, on the other hand, when our cells are getting ready to go through some kind of cell division, our cells use special machinery, special enzymes that package this DNA up into these chromosome structures. You can see how this chromatin is being coiled tighter and tighter and tighter. They're kind of organized into this like flower pattern, these loops, until eventually we have this much larger and visible structure. Here's an image of a replicated chromosome. Now, on the other hand, if we do look at a cell underneath a microscope and the DNA of the cell is actually in its chromosome form, we can see those chromosomes, and we will see them in lab coming up shortly. Now let's talk about a chromosome a little bit. This image here shows you what we call a replicated chromosome. We only have chromosomes inside of a cell after DNA replication has occurred. And what we've got going on here is basically we have this structure where we have this half of the chromosome here on the left, and we have this half of the chromosome here on the right. We call those two parts, we call them sister chromatids. And what's really going on here is we basically have information and we have a copy of that information. We have two sets of the exact same information held together by something known as a centromere here in the center. And there's an enlargement of what we've got going on inside of the nucleus. The information on these two halves, on these two sister chromatids, is exactly the same. Not similar, it is entirely the same. What I mean by that is if in this particular location right here, we have a gene, and let's just say that that gene contains the information for eye color, and it specifically has the information for brown eyes, at the exact same location on the other sister chromatid, we will find the exact same recipe. Not similar, not blue eyes, brown eye information will be found on the sister chromatid. Because the DNA recipes that we find on these two molecules of DNA, since they are identical, we call them sister chromatids. We never have a situation where we have DNA on one sister chromatid that's genetically different from DNA on the other sister chromatid. That's not how our cells work, and that's not how mitosis works. Once again, though, we do have a structure called a centromere that's holding the sister chromatids together. I think I pretty much covered these ideas, but again, normally inside of a cell, we don't have DNA in chromosome form. Instead, we have DNA in chromatin form. When the DNA is in chromatin form, the recipes are accessible. The cell can go through transcription and make messenger RNA from a particular gene. That messenger RNA can be translated by a ribosome and a protein can be produced. Genes can be turned on and off when the DNA is in chromatin form. However, when the cell is going to begin the process of mitosis, the DNA, it had to replicate. And then after replication, if everything seems to be going fine in the cell, there's no major problems with the cell, then the cell will start the mitosis process. And as it begins the process of mitosis, the DNA condenses, it winds up, and it takes on that chromosome highly organized form. Some other images here. We've talked about how we have 46 chromosomes inside of our cells. What we basically have is we have 2 pairs of chromosomes, or we have a pair of chromosomes for every kind of chromosome that is inside of our body. We have 23 pair of chromosomes. 23 chromosomes we inherited from our biological mother, and 23 chromosomes we inherited from our biological father. So we each inherited maybe like a chromosome number 1 from our father, as well as a chromosome number 1 from our mother. Now, these chromosome number 1s, we call them homologous. Homologous chromosomes are chromosomes that have the same kinds of genes on them. But because these chromosomes are actually inherited from 2 different people, the specific gene sequences, we call those alleles, A-L-L-E-L-E-S, alleles. The specific versions of the genes or alleles could be different. So from our last example, again, talking about sister chromatids on a particular chromosome, maybe we have over here a gene for eye color. And we have specifically the recipe for brown eyes at this particular location here on this gene, on this chromosome. Right here, we have information for brown eyes. At that exact same location on the other sister chromatid, we have the information for brown eyes. Again, it would have to be brown eyes, recipes for brown eyes on both of these sister chromatids because of the process of semi-conservative replication. We understand that through semi-conservative replication, we have an old strand and a new strand making up one molecule of DNA and an old and a new strand making up the second molecule of DNA. Well, what's going on here is basically we have one strand of DNA that has condensed. That strand has old and new parts on it here. Here's your copy with old and new parts on the other sister chromatid. Going back to our eye color example, again, we have information for brown eyes here, but because the other chromosome is from a different person, while there is information about eye color at the exact same location on this arm of this chromosome, the information might actually be a little bit different. The recipe could result in a slightly different amount of the protein being produced, or maybe even a different protein. Maybe here we have information for blue eyes. Again, these chromosomes are homologous because of the fact that both these chromosomes have information for eye color at the exact same location on each chromosome. But again, because the blue chromosome was actually present in the sperm and came from dad, and the red chromosome was present in the egg and came from mom, and since they're not related to each other, hopefully, we might have different specific recipes on these 2 chromosomes. Yet, the chromosomes are homologous. What we probably have here on the rest of the chromosomes is we have, again, the exact same genes and exact same recipes on the father's chromosomes. We have same genes, but possibly different recipes on the mom's chromosome. The mom's 2 chromatids are sister chromatids to each other, but they are not sister chromatids to the father's chromosome. We'll have more practice with that, but just introducing that concept at this point. All right. It's time for the cell cycle. So now that we've talked about bacteria,